Right, here we go. So in our previous lecture, I talked about, well, we finished talking about symbiosis, and I talked about the most likely scenario for the evolution of early eukaryotes. This would have been roughly 2.1 billion years ago. We've got, by the way, some evidence from geology that that is about the time when the Earth's atmosphere crossed what's called the Pasteur point of 1% of present day oxygen levels. There wasn't any oxygen in the earliest Earth's atmosphere because oxygen is so reactive. It would react with things like dissolved iron in the oceans and wouldn't stay in the atmosphere in the gaseous state. What put it in there was the activity of blue-green bacteria, basically. And once the oxygen levels crossed 1%, that's enough for aerobic life to survive. You couldn't, but there are aerobic organisms that could. In fact, what may have driven the evolution of eukaryotes is the fact that oxygen is deadly poisonous to so many microbes, to these anaerobes, because it's highly reactive. You know, somebody pointed out that cosmically having life that requires oxygen is kind of like a life form that must drink gasoline in order to survive. It's a very dangerous chemical for most microbes. And what may have driven eukaryote evolution is the symbiosis may have appeared um, as a means of coping with the increasingly high levels of deadly, dangerous oxygen in the atmosphere. It's called the ox-tox hypothesis. You can look into that if you want to. That's why the gods gave us Wikipedia. But eukaryotes are now here. And the latest scheme uses two humongous supergroups. There's also, well, the excavata don't clearly fit in either one, and we're not entirely certain that they are monophyletic. That still hasn't been resolved. And there's a few funny little lineages that don't go anywhere, that we're not sure where they go, or they don't fit into one of the two neatly. Um, most of them are not critters you're ever likely to encounter unless you really deliberately go out and look for them. But for the purposes of this class, we'll use these two supergroups, the Diaphoretickes and the Amorphia. And then the excavata might be a third group, or they might go in one of the two, and they might not be. Okay. And I'll start with the Diaphoretickes, Haptista, Archaeplastida, and then Sar. I'm not going to say much about Haptista, and I'm not going to say much about Archaeplastida. Archaeplastida includes plants, green algae, red algae. SAR is a very prominent group, and SAR stands for Straminopiles, alveolates, and Rhizaria. And I may not get to the alveolates here, but I'm going to cover as much of this as I can today, and we'll see how it goes. Okay. The thing about Haptista and Archaeplastida is that a lot of them are photosynthetic, and as such would typically be covered in classes on botany. Now, that whole everything is an animal or a plant dichotomy is, 18th century BS. And in fact, there are photosynthetic critters all over the tree of eukaryotes, and there are animal-like critters all over the tree of eukaryotes. But still, I'm going to leave Dr. Noyes something to cover, and I won't say too much about the more photosynthetic groups that we're talking about. Haptista, you've got two major groups of those. Centrohelids are cells that have round cell bodies and long ray-like extensions. Protists with this shape used to be called heliozoans, which is Greek for sun animals. because they look like the drawings of the sun that you made in kindergarten, a circle with lines radiating from the circle. They're all microscopic, by the way. The ray-like extensions are supported by internal thick bundles of microtubules, and they're called axopods. And unfortunately, for people who like things neat and simple, centrohelids go in the haptista, but there are other protists in other lineages that have convergently evolved this basic shape. So just because we have to make things confusing. So yeah, this is the set of all protists that have axopods is pretty polyphyletic. It's evolved independently in several different groups. Many centrohelids, by the way, also have little silica scales on the cell body. And then the second major group, the Haptophyta, they're photosynthetic. They don't have rays. Many, although not all, are covered in scales made out of calcium carbonate called coccoliths. And cells that carry coccoliths are sometimes referred to as coccolithophores. By the way, I will never test you on the meaning of Greek and Latin words, but some people find that learning the roots can help with vocabulary, not just in this class, but in many others. That for right there is the Greek root for to carry. So coccolithophores just means things that carry coccoliths. Lithos is Greek for stone, and kokkos just means something that's round. So these are the round stone carriers. There are some haptophytes that are naked and some have silica scales, but the best known are the ones that carry these calcium carbonate coccoliths. And here's what they look like. On the left, that's a centrohelid. It has got silica scales. You can't really clearly see them here, but you can see it's got this round cell body and then these long ray extensions coming out. The thing can actually do phagocytosis on the rays. And if you look very carefully and squint and probably take a lot of drugs, you can see little vesicles that are on the rays that travel up and down, a little bit like elevators on a shaft. This is actually an interesting model organism for looking at vesicle transport. On the right, there's a haptophyte, and the cell body is round. There aren't any rays, and the cell is covered in these coccoliths. Notice the scale. The coccoliths are only about 2 microns wide. They're not very large, but they do have the really interesting, complicated shape to them. And most species have coccoliths that have very distinctive shapes. There's lots of different diverse varieties of coccoliths. I wouldn't go into much detail about haptophytes, except that they are some of the most abundant eukaryotes on the planet. There are haptophyte blooms. This one was photographed in the English Channel. This is the southwest tip of England. This thing right here, by the way, is the city of Plymouth. And yes, that's where the pilgrims set forth on their journey to America. And that's why the place where they landed is called Plymouth Rock. This peninsula is the county of Cornwall. And in the English Channel, all of this light turquoise swirly stuff is a massive bloom of haptophytes. They're extremely common. There are parts of the ocean where they make up something like 50% of the primary production. They live in huge numbers. They get fed upon in huge numbers. As they live and as they die and as they sink to the bottom, they are one of the main pumps that takes carbon dioxide gas out of the carbon cycle. Okay, the carbon cycle is, plants take up carbon dioxide, consumers eat plants and breathe out carbon dioxide. So there's carbon dioxide gas in the atmosphere. We're putting extra carbon dioxide gas in by burning fossil fuels. One of the things that takes it out of circulation is that organisms that make calcium carbonate minerals, the carbonate ion has to come from somewhere, and it comes from dissolved CO2. And as these things make their tiny little calcium carbonate scales, they live, they die, they sink to the bottom, and they build up huge layers of soft calcium carbonate mud. And when they end up on the bottom of the ocean, those layers of calcium carbonate mud made of untold cajillions of little coccoliths ultimately pack down into a rock known as chalk. And this is the chalk layer that makes up the White Cliffs of Dover on the southern coast of England. It is, I think, about 45, 50 million years old. I'd have to look up the exact date. But it is made mostly of fossil coccoliths that lived in humongous numbers, died, sank to the bottom, and then ultimately got uplifted by plate tectonic forces to produce this rock layer. Much the same is true, by the way, for there are chalks in Arkansas. There's a layer in the southwest called the Annona chalk that I've actually poked around in, and I've looked at pieces under our SEM, and I can confirm that it's mostly made up of coccoliths. So these are extremely important group, from a global perspective in terms of their enormous abundance and their importance in building up rock layers and in the global carbon cycle. So there. So that's all I'm going to say about the Haptista. The Archaeplastida includes land plants, green algae, red algae, and a couple of minor groups that all form a single clade. Sometimes you'll see that clade called plantae, but I'm using Archaeplastida. Archae, of course, means old, and plastids are plastids, like chloroplasts. And it is thought that this is the only branch of the eukaryote tree that formed a primary symbiosis with blue-green bacteria. The chloroplasts are only surrounded by two membranes, and in an early ancestor of the Archaeplastida, this is the group that engulfed a blue-green bacterium or cyanobacterium and established that symbiosis that led to chloroplasts. There are lots of algae that don't belong in the Archaeplastida. They've got chloroplasts surrounded by 3 or 4 membranes, and there's evidence that they borrowed their chloroplasts from Archaeoplastida. This is actually really weird. There are some protists whose ancestors evidently engulfed a green alga in the Archaeoplastida and digested most of it, but kept the chloroplasts. That's called kleptoplasty, and it's surprisingly common. There are others where the chloroplasts are actually remnants of a complete archaeoplastid alga that got engulfed. So you've actually got some photosynthetic protists that have symbiotic archaeplastids inside them. And if you look at how different groups of protists acquired the ability to photosynthesize, it starts looking like the plot of a heist movie, because different groups have either stolen chloroplast from others, or their chloroplasts are actually complete eukaryotes that they engulfed. So chloroplasts get passed around all kinds of crazy ways. I'll show you a really kooky example of that probably next week. There's a small group of archaeplastids called the glaucophytes. Glaucos, by the way, is Greek for a color that encompassed what we'd now call light green, light blue, and gray. So these are the gray, blue, green algae. And I mention them because their chloroplasts still have bacteria type cell walls. So their chloroplasts retain a feature of the cyanobacteria that they evolved from. That feature's been lost in other algae and in land plants. You won't find bacteria type cell walls if you look at the chloroplasts of a land plant, but they're still around in these glaucophytes, which are otherwise very small, single-celled, free-swimming eukaryotes. One major lineage of the archaeplastids are the red algae. They look red because they've got accessory pigments called phycobiloproteins, and they've got chlorophyll A. There's several types of chlorophyll. Let me see. The red algae that you might know best would be, I think, the nori algae that are eaten in Japanese cuisine, notably in sushi. If you get sushi rolls wrapped in seaweed, it's actually a red alga. There's another red alga that is commercially harvested in Europe, and there's a product made from it called carrageenan, which has kind of a creamy texture, and it's the sort of thing used to add creaminess to things like artificial diet foods, like low-calorie ice cream, where carrageenan might be used to take the place of the butter fat that makes full-fat ice cream creamy. So those are the red algae that you're likeliest to encounter, I suppose, in daily life. The other major plantae lineage is sometimes called viridiplantae, and that's green algae, like this single-celled plant called a desmid, particularly beautiful, I think, green algae. And then the land plants, which evolved from green algal ancestors, and they use two types of chlorophyll, chlorophyll A and chlorophyll B. And if they run out, they get more at a chlorophyllling station. Ah. Okay, I'll give you a few seconds to pick yourself off the floor because I know you're all just paralyzed with laughter at my joke. Right. So that's all I'm going to say about the Archaeplastida. If you really need to know more about them, we do offer, Dr. Noyes does teach biology of lower plants and fungi. But I'm going to leave that there and move on to SAR, which is a humongous monophyletic group that's called SAR because it includes three major subgroups: stramonophiles, alveolates, and rhizaria. Stramonophiles includes an awful lot of photosynthetic algae and also some non-photosynthetic groups, and I'm not going to have much to say about the stramonophiles here. Alveolates is really big because the alveolates include ciliated protists, a group called dinoflagellates, and a group called apicomplexans, which includes some of the most important parasites. If you took parasitology with me, hopefully you still remember the apicomplexans in agonizing detail. And then finally, rhizaria. There's a lot of weird rhizarians, but if there's a common feature to them, they are vaguely amoeba-like protists with long, thin extensions of the cell body, pseudopods, often supported by branches of the cytoskeleton. A number of important rhizarians form skeletons, and as such, they've got a very long fossil record. So moving on to SAR, straminopiles includes everything from unicellular photosynthetic forms like diatoms. This is a diatom over here on the left. It's accessory pigments. It's photosynthetic. It's got chlorophyll, but it's got extra pigments that give it this golden brown color. And it's got this very regular geometric shape because diatoms secrete um, uh, exoskeletons made out of silica that have this very regular and very detailed shape to them. Diatoms, by the way, are abundant in both most freshwater ecosystems and in the oceans. They're very important as primary producers. And that's just a unicellular critter that might be maybe 50 microns long. But the straminopiles also includes multicellular forms like this bull kelp, which can reach, I think, over 100 feet in places like the kelp forests off the coast of Central California. I've seen these things wash up on the beach and they can get absolutely huge. There's a lot of non-photosynthetic protists in the straminopiles, and some of them are superficially similar to fungi and used to be classed as fungi. Possibly the most notorious is this thing. This is a plant parasite called Phytophthora infestans. It is a parasite of, I think, over 300 crops. It is really not something you want to see in your fields if you're a farmer, if you're a vegetable grower, especially. And it was the cause of Angorta mor, which is the Irish term, means the great dying, and it's the term for the potato famine of 1845 to 1850. Ireland had a very large dispossessed population living on the margins, surviving mostly on the potatoes they could grow. And potatoes are a good staple crop because they can grow in poor soil and they're nutritious enough that you can sustain life on a diet of mostly potatoes. It's not that much fun, but you can live on them. And in 1845, Phytophthora infestans attacked the Irish potato crop, which was pretty much all one variety of potato. So it wasn't very genetically diverse. And the upshot was about 1 million Irish people starved to death, and roughly another 1.5 million emigrated. Many of them came to the United States. And if you've got Irish ancestry, it's certainly not the only time that the Irish have immigrated to the United States, but your ancestors might well have come as a direct result of the potato blight. Phytophthora infestans might well be the reason why your ancestors came here. Other straminopiles that are fungus-like include the so-called oomycetes, commonly known as water molds. They're important decomposers in freshwater ecosystems, and some of them cause disease in plants. Some of them cause disease in critters like fish. So very diverse group, the straminopiles. A lot of these sun animals, heliozoans, sun is helios, zoa, animal, are in the straminopiles. This is one of them. And yes, this does look an awful lot like the one that we've already seen, but it's not closely related. As I was saying, that sun-like shape with that round central body and axopods sticking out of the center, that's evolved convergently. That's evolved several times. This species, by the way, also just happened to have single-celled green algae as endosymbionts. So that's what the green spots in there are. And there's a cross-section through an axopod, Axopods are supported by these big bundles of microtubules, and you can see this is a rather complicated arrangement. And surrounding this bundle of microtubules, you can see various little vesicles up there and up there and up there. And as I mentioned before, the cytoplasm surrounding these microtubular bundles can do phagocytosis, and it can engulf and entrap tiny food particles. And vacuoles with food particles can slide down an axopod to the main cell body where they get digested. And I think I mentioned that critters like this have been used as model organisms in study of vesicular transport in eukaryotes. You can see this happening under a decent light microscope. Okay, right. So what makes all of these straminopiles as crazy diverse in shape as they are, everything from things that look like fungi to sun animalcules to bull kelp, what makes them all straminopiles is that if they have a flagellated stage, um, not all of them do, but many of them do at least part of their life cycle. Typically, it will have two flagella. One of them, the one on the left, is smooth. That's this one. And the other one has little branches from it, which makes it look like a piece of Christmas tinsel. These little hairs are called mastigonemes. And this is what gives the clade its name. Stramen is Latin for straw. So straminopiles means it's got extensions from the cell that look like little pieces of straw sticking out. So straminopiles are defined on the basis of similarities in their DNA. They're defined by those DNA uh, similarities that are discovered through cladistics or other type of phylogenetic analysis, like we talked about. But members of the straminopiles, if they have a flagellated stage, it very usually looks like this, where you have one of the flagella with these little mastigonemes branching from it, making it look like tinsel. Right. That's all I'm going to have time for in here about these straminopiles. And again, if you want to know more about them, I'm going to save something for Dr. Noyes to cover in his lower plants and fungi course. I'm not sure when he's offering that, but it's a good one to take if you want to get the full spectrum of eukaryote diversity. Rhizaria. They're not closely related to the amoebozoa, which are where we put a lot of the amoebas, but they are amoeba-like protists in that the cell itself doesn't have a fixed shape. In many rhizarian groups, there's a specialized cytoskeleton of microtubules that supports axopods. We've seen these before, and again, this shape of a round cell body with axopods sticking out like sun rays has evolved independently at least 3 times and probably more. But there are other rhizarians that don't have axopods, so it's not a defining feature of the group. The ones we'll focus on are the ones that produce hard skeletons, and both of these groups have a fossil record going back over 500 million years. The first is Radiolaria. These have a complex internal skeleton made of silica, or there's actually one funny little subgroup in which the skeleton is not silica, it's strontium sulfate. The only group I'm aware of that uses strontium as a biomineral. And in Rhizaria, there's a clear difference. There's an inner cytoplasm called the endoplasm that often looks very dark. That's where the organelles and things are kept, and the nucleus. And the outer cytoplasm is full of vacuoles, so much so that it looks like foam. The two are separated by a membrane structure called the capsule, and the skeleton of silica typically forms on the capsule. That sounds weird, but I'll show you one and you'll see what I mean. Foraminiferida, aka forams, they don't have axopods. Radiolarians do. But the pseudopods, the extensions of the cell, are very thin and they're very flexible and they form a network. They don't just branch, they branch and they rejoin. These are called reticulopodia. Rete is Latin for a net, and a reticulum is a little net. So these pseudopodia, these cell extensions, form a network of reticulopodia because they don't just branch, they come together as well. Most species, not all, but most, form tests of organic matter. And I think I said this before, but I'll mention it again. Testa is Latin for a pot. And in protists, critters that make hard structures that look somewhat like pots, those structures are often called tests. You'll see this in more than one group. It's got nothing to do with exams. It's just comes from the Latin word for a pot. So most forams produce tests of organic matter, or organic matter that glues together tiny grains of minerals that the forams pick up. We call this being agglutinated, by the way. An agglutinated test is a test made out of tiny grains of minerals that have been glued together, or many of them, including the ones we're going to see in lab tomorrow and Monday. The tests are made of calcium carbonate. And the tests can be just single chamber, just like a vase. But in many species, the tests are multi-chambered and they can take all kinds of very complicated shapes. And then finally, cercozoa is what we'll use for rhizaria that aren't radiolarians or forams. Some of these are amoeba-shaped, some of them aren't. Some of them make tests, like a group called the phyllost-testate amoebas. Some don't. It's a pretty diverse grouping. So phyllost-testate amoebas, I always have to mention testate amoebas because the professor that I did my undergraduate honors thesis under back in 1990, 91, back when hair metal was still a thing. So I always have to give a shout out to testate amoebas. So thank you, Dr. Bamforth, wherever you are. These have very fine, slender pseudopods instead of the blobby or lobopseudopods of the amoebozoans, which include other testate amoebas. We'll get to those later. Many of these have tests made out of silica scales that they secrete internally in a big vacuole and then export to the outside. So if you look at this picture under the electron microscope, this thing looks like a pine cone. You can see it's made up of tiny scales. And the scales actually overlap. They're kind of shingled together to make this vase-like structure. So that's what one looks like under the scanning electron microscope, and that's what one looks like under the light microscope. And these are pretty common in pond water. They're pretty easy to find if you've got enough patience. There's other cercozoans that are just small and flagellated. There's other cercozoans that include this really weird protist of amoebas where you have multiple cells that join together in a network and they've got green algae living inside them. This is the weird marine multicellular amoeba chlorarachnion. So lots of cercozoans out there. Cercozoans are weird. There's a living radiolarian. And what I want you to see is, first of all, the dark area at the center is the endoplasm, and the nucleus is at the very center of this. And surrounding it is where most of the organelles are. And the ectoplasm is this zone around here, and you can't really make out any detail of it. And it looks very light, and that's because it is so full of vacuoles that it basically looks like froth. You know, it looks like the froth on top of your espresso at Starbucks. And coming out of it, you can see, okay, first of all, the capsule is a membrane structure that is the boundary between the endoplasm and the ectoplasm. You can't really see that it is a membrane, but it is located right there where I'm drawing it. And you can see axopods sticking out of this thing. And incidentally, if you're curious as to why it has them, radiolarians are not all, but many of them are planktonic. Plankton, by the way, means that you live floating in the water and you're not a strong enough swimmer to control your position in the water. Plankton floats and has to go where the current goes. These are planktonic. They'll feed on tiny bacteria and things like that, trapping them with their axopods. And a shape like this slows down the rate of sinking, for much the same reason that, you know, things like milkweed fluff, milkweed seeds have the shape that they do. You've ever seen one of those, a thistle seed or a milkweed seed, or heck, even a dandelion seed has all of those hairs on it, and it makes it possible for those things to float in the lightest breeze. They sink really, really slowly. It's pretty much the same reason for radiolarians and for those other heliozoans that we've talked about. So that's what a rad looks like. And you might notice that some of these things that are sticking out look to be a lot spikier and thicker, like that and that and that. And that's because rads deposit silica on the capsule, and typically they'll have rays made of solid silica that stick out. And if we took this radiolarian and removed all of its cytoplasm, it would look like this. That's the silica skeleton of a radiolarian that I don't think is hexacontium, but it is closely related to it. And you can see you've got this ball right here with this very geometric, almost soccer ball pattern on it. That forms right on the capsule. And you've got various rays and spines of solid silica sticking off that central capsule. So some of these are just absolutely gorgeous. They look like Christmas tree ornaments. But they're found in the oceans, sometimes in very great numbers. Many rads are spherically symmetrical, like the one you just saw, but in one major group called Nassolarians, instead of having spherical skeletons, they've got skeletons that are cone-shaped or possibly helmet-shaped. Like Lamprocyclus martialis on the left looks like the kind of heliton that, Gozor, the dark galactic lord, would wear on his head. So here we'll put in an eye, and there's the other eye, and there we have the mouth of Gozor, the galactic overlord, in his ceremonial helmet of cruelty. And then there's a species on the right that I'm not sure what that is, that's a little bit less spiky. But yeah, some of these have skeletons that are more cone-shaped or helmet-shaped. And that's a foram, or a foraminiferan, alive. And the test is this brown thing right here. The bulk of the cell is going to be in there. The nuclei are in there. These guys have multiple nuclei per cell, by the way. And most of the organelles would be there. And then extending away from the cell body inside the test is this network of very, very fine extensions that branch and rejoin. So those are the reticulopodia that make up that little net. Forams, some forams have algae living inside. They have endosymbionts. But most forams trap smaller organisms with their reticulopods. They'll just pick up things like bacteria or single-celled algae or tiny bits of detritus, trap them with the reticulopods, and transport them to the main cell body. So that's what a foram looks like. And the tests can have all kinds of awesome shapes. The one down here is called monothalamic. It's got a single chamber. The chamber just happens to be coiled like a snail shell. And by the way, some of the earliest people to study forams thought that they were related to the mollusks, to the snails or to the squid, because of the shape of their test. They're not. It's convergence again. These others, however, have multiple chambers. So that one right here, you have multiple chambers in a spiral. Here you've got multiple chambers in an alternating branching sequence. And there are plenty that have even kookier shapes than this. Most species have calcium carbonate tests. There are some where the test is just tough organic matter, something like chitin, although not the same. And many of them have agglutinated tests that are made of little sediment grains glued together. By the way, that foram, by the way, a foramen is Latin for a window. Foramina is the plural, foramina. If any of you clowns get into medical school, you'll have to learn a bunch of foramina. The base of your skull, for example, has multiple foramina. They're the holes by which your spinal cord and your cranial nerves come out. So the foramen magnum is the big window by which your spinal cord comes out of your skull. And you've got the foramen ovale and the foramen lacerum and the foramen rotundum. And I used to know what these did, but I haven't taught gross anatomy for maybe 30 years. But those are the windows in your skull that the nerves pass out of. So you'll see this root foramina before. And the fera is Latin for to carry. So foraminifera are the things that carry many windows. And this is what that means. This is a close-up of a single foram test. And this, by the way, is a species that is planktonic, that floats. The ones that you saw earlier are all species that live on the bottom. They're benthic, as we say. But this is a floating one. And you can see that the test is full of tiny little holes. You couldn't really see those on this slide at this magnification. But if you were to zoom in, you would see that the whole thing is perforated with thousands of tiny little holes. And it's these holes that the reticulopods come out of those branching and rejoining extensions of the cell. They come out of the foramina. So these holes are the foramina, and a critter that has lots of foramina in its test is a foraminifera, a carrier of many windows. So there. Yeah, when you see these guys up close, the tests look like they've been hit with a shotgun. They've got tons of foramina there. That's a planktonic foram. This is one of the floaters, and in life, you can see all those reticulopods coming out. And these do the same thing that the axopods do in some of the other protists that we've talked about. They enable a planktonic foram to float by slowing down its sinking rate. And again, this is the same evolutionary logic behind the very fine hairs on a milkweed seed or a thistle seed or a dandelion seed. All of those hairs slow down the sinking rate and make it possible for one of these seeds to drift a long distance if there's any amount of wind at all. So planktonic forams float by means of the reticulopods, and they also trap food particles. And this is how abundant forams can be. There are forams that can be quite large. They range from maybe grain of sand size to these particular ones get up to the size of a dime. They're quite readily visible under to the naked eye. And this genus Nummulites, it's a foram. You can see traces of the multiple chambers that it has. These are fossils. They're about 40 million years old, and they make up quite a lot of the limestone deposits in parts of Egypt, the limestone that was quarried to build the pyramids. The pyramids used to have polished marble on the outside, although that has almost all gotten stripped off. But the core of the pyramids is made of blocks of limestone, which is relatively soft. It's easy to quarry, even with stone tools. And it happened to be located very close to where the pyramids were built. The quarries are still around. We still have the places where all those blocks were dug up. And that limestone that the pyramids are made of is made up mostly of these forams, Nummulites. The ancient Greeks actually noticed them, and the legend was that they were the petrified remains of the lentils in the pyramid builder's lunch. That is, they were sloppy eaters. They dropped some of their lentils, and they must have hardened into rock. But in fact, they are what makes up that limestone itself, and they're fossil forams. So yeah, if you ever see the Great Pyramids of Egypt, they're mostly made out of forams. Because forams have such a long fossil record, and there's so many darn species of them, they're also used extensively by paleontologists, in particular in the oil and gas industry. One of the few fossil specialists who ever died wealthy was a guy named Joseph Cushman, who figured out how to use the appearance and disappearance of different species of foram in the fossil record over time as a tool for working out, how deep your oil well went and how much deeper you might have to go to strike oil, and became a consultant for the petroleum industry round about the year 1900. And in fact, most paleontologists until recently were employed not by museums, but by the oil and gas industry and specialized in things like forams. Dinosaurs are great, but this is what pays the bills. Sizable portions of the deep ocean floor are covered in mud made of dead foram tests. Now, I've mentioned that this happens with coccolithophorids. There's areas that are covered in dead radiolarian skeletons, and there's areas of the ocean that are covered in dead foram skeletons. This is called foram ooze. Given enough time, this will eventually compact into limestone. And if it hardens into limestone and ultimately gets lifted up by plate tectonics, in a few million years, somebody might build pyramids out of this stuff. That photo was taken by a famous research vessel, a submersible uh, used for deep sea exploration called the Alvin. So yeah, that's dead foram ooze. In fact, forams are so awesome that there's actually a sculpture garden in a city whose name I probably won't pronounce right, but I think it's Chongshan, close to Hong Kong in China, because a local marine biologist convinced the local authorities that what they really needed was a sculpture garden with sculptures of forams, and convinced the authorities to build it. So you can go walking through Foram Park if you're ever in this part of China. And you can see a lot of these things have very complicated and strange and in a way very beautiful shapes of their tests. There's lots of different varieties, and many of them are aesthetically very pleasing. My major professor in grad school was a foram specialist, and he used to say they were the best things to study because they're found in oceans all over the world, none really in freshwater, but they're found in oceans from the Arctic to the tropics to the Antarctic. They're found in everywhere from the surface waters to great depths. And they've also got a very extensive fossil record on all continents. So if you study forams, you can write grants to do research pretty much anywhere you want to go. My major professor had studied forams in the Antarctic and in the tropics and in something like 150 countries. He had a huge number of places that he'd been able to visit because of his studies of forams. That was back when you could get grant money, but it's a little harder to do now. But they still provided for him an exceptional career. Right. Okay, so we're still talking about SAR, and I've said all I'm going to about the Straminopiles, and I've said all I'm going to about the Rhizaria. The third group, the A in SAR, is the alveolates. And what links these is they've all got a set of flat vesicles directly underneath the plasma membrane, the external membrane of the cell. The effect is a little bit like quilting. And these set of vesicles are called the alveolar membrane system. And the three members of the alveolates don't look very much alike when you see them under the microscope, but they've all got this alveolar membrane system. And that, aside from DNA sequences, is the evidence that we group them together as one clade within SAR. And so the members of alveolates include Ciliophora, the ciliated protists, Apicomplexa, which is one of the major groups of parasites. Apicomplexans are exclusively parasitic, and they include, probably the most notorious is Plasmodium, which causes human malaria and kills something on the order of 700,000 people every year. One of the big global public health problems. Apicomplexa also include things like coccidians, which cause disease of dogs, cats, livestock, poultry. Anybody who has worked in a vet's office or is thinking about going to vet school you'll see a lot of coccidiosis. Those are in the apicomplexa as well. And then finally, dinoflagellates are one of the main groups of primary producers in the oceans. We also have them in freshwater. Population explosions in the ocean of dinoflagellates cause what we call red tides, which can be catastrophic for the local ecosystem. So, very diverse group, three major players in it, but they're all grouped together in the alveolata because they've got these alveolar sacs under the plasma membrane. There are specialized alveolin proteins in those membrane sacs, and here they have been stained with green fluorescence. So the green in the pictures above marks where these alveolar sacs are located. And in these dinoflagellates, you can see that they form this layer right around the perimeter of the cell. In Plasmodium, this is the malaria parasite, and they form that layer outside the cell. And Paramecium is a ciliated protist. It's one you may have seen. And they form that layer right around the outside of the cell, just underneath the external membrane. By the way, the nuclei are what have stained blue. So there we've got this link among these three taxa that are otherwise so very different from each other. And we actually ended early because that's where I decided to close, because if I start with any of these three groups in the alveolata, I'm not going to finish. So I'm going to go ahead and bless this and call it dinner, and I'm going to stop the recording now.